In Chapter 25, we will be briefly looking at amino acids. So what you are seeing here is a hand with fingerprints. A fingerprint is often the most important piece of evidence left behind at the scene of a crime. Forensic scientists use a lot of different methods to visualize fingerprints. One of them involves the use of a chemical agent called ninhydrin. This reacts with amino acids present in fingerprints, and that will produce a colored compound that can be visually observed, as you can see there. What are amino acids and why are they present in our fingertips? What functions do they serve? There are 20 naturally occurring amino acids, and they are the building blocks of proteins. The functional groups are separated by only one single alpha carbon, which is why it's called an alpha amino acid. So this is the alpha proton of the alpha carbon, and on that carbon we have an amine group, a carboxylic acid, and a side chain, which is symbolized by R. A peptide is a short chain of amino acids, usually 40 to 50, or it can be less than 50. A dipeptide is 2 amino acids. A polypeptide would be 2 to 39 amino acids. And proteins are greater than 50 amino acids. And can be up to thousands. Most people's definition of protein is something that you're gaining in your diet via meat, beans, tofu, something like that. But proteins in chemistry, there's always a chemistry definition, is a lot of amino acids strung together. So each amino acid has a carboxyl, an amino group, side chain, and that alpha carbon. So those are the 4 parts of an amino acid. And there's 4 different ways to categorize amino acids, and it's classified by its side chain. So the first group are made up of non-polar side chains. You do not need to memorize amino acids for this class. I'll let whoever's teaching biochemistry take care of that portion for you. We're just introducing each of the structures of the amino acids. So amino acids have a name, they have a side chain, there's a 3-letter abbreviation as well as a 1-letter abbreviation. The first amino acid here is glycine, abbreviated GLY and G. Glycine has the side chain that is H only, and this is the only amino acid that is achiral. And that's because there's a H as a side chain, so there's 2 hydrogens on that carbon. The next amino acid is alanine, which has a CH3 for a side chain. This is ALA for 3-letter, and then A for the 1-letter abbreviation. The one with the isopropyl as the side chain is valine, Val and V. This one is called leucine. I don't understand why, and you'll understand why I don't understand why when I show you the next one. This is L. So leucine has an isobutyl group. This one is isoleucine. So it's counterintuitive. I would think that the one that is isobutyl would be isoleucine, but it's backwards. So isoleucine, this is a sec-butyl. The abbreviation here is ILE, and I is the 1-letter abbreviation there. One of the sulfur-containing amino acids is methionine. MET is the 3-letter abbreviation, and just M is the 1-letter abbreviation. Proline is the only amino acid that has a cyclic structure that's part of the side chain. And that's P. And this one is called phenylalanine. So there's the phenyl group and a CH3 for the alanine. So this one is phenylalanine. PHE. And since there was already a 1-letter abbreviation of P, phenylalanine sounds like it starts with an F, so F is the letter for phenylalanine. This one here with the indole ring There's a CH2 indole. This is tryptophan, the amino acid accused of the cause of heavy napping on Thanksgiving. This one is abbreviated TRP. And the one-letter abbreviation is W because doesn't everybody call it tryptophan? Okay, W, tryptophan, W. So this amino acid only causes sleepiness if you eat large quantities of it. You would probably have to eat several turkeys at Thanksgiving in order for it to make you sleepy. The truth behind drowsiness at Thanksgiving is excessive eating. Next Thanksgiving, you can share that nugget of information with your family if they start talking about tryptophan. The next category of amino acid side chains are polar ones. So these are polar and they're also neutral, so there's no charge to these side chains in their current form. The first one we have is asparagine. It's abbreviated ASN for asparagine. So that means the one letter is not A because we use that for alanine. This one is N for the end of asparagine. That has an amide group, so there's a CH2 amide. If you add an extra CH2 spacer to that, that makes it glutamine, which is abbreviated GLN, and a Q because you can think of it as cutamine. So that is that one. A CH2OH, an alcohol one, this is serine. That one is SER and S. Serine is one of the ones that is used a lot of times in the catalytic triad. In a lot of enzyme functions, we see serine. If you have the secondary alcohol, this is threonine, THR, and this one gets the T. And because that one gets the T, this next one here does not get the T. This is tyrosine. TYR. So this has a CH2 with phenol on it, and this is Y from tyrosine. Our other sulfur-containing amino acid, there's only two of them. This one is cysteine, CYS, and C. And here we have acidic and basic side chains. These are polar and acidic. The first one here is aspartic acid. This one looks like asparagine from the last one, but instead of having the amide, there's the carboxylic acid on the end. This one is asp. Letter is D, because you can think of it as aspartic acid. This one is glutamic acid. So similar to glutamine, but now there's a carboxylic acid instead of the amine there. This is glue, and that one is E because we're running out of letters. There's no other things to remember that one other than we're running out of letters. Amino acids with basic side chains. This one is lysine, which has 4 CH2s and the amine group. That is lysine. And L was taken, so the next letter available was K. This one is histidine, and it has that imidazole ring on there. This one amino acid is also used a lot of times in the catalytic triad because it reacts as a base, so it can pull protons off of different amino acids. And this last one here is arginine. That one is Arg. This is also the pirate's favorite amino acid, and that's R is the one letter abbreviation there. This group here is called a guanidine. So we have carbon double bond N to 2 other N's. So those are our 20 naturally occurring amino acids. Our bodies can make 10 of the 20 amino acids that we need, but we have to get the 10 other amino acids from our diet. So meat eaters can get all of their essential proteins from meat, fish, milk, and eggs, but there's not a single plant source for vegetarians to get all of their essential amino acids. So if you're a vegetarian or a vegan, you'll need to consume a combination of grains, beans, and nuts in order to get those extra amino acids, or you can take a multivitamin. With 20 amino acids and 20 different side chains, each of the amino acids and their side chains are going to have different pKa's. So under acidic conditions, we are going to see the amino acids fully protonated, which would be here. This is acidic. It's fully protonated. So our carboxylic acid is protonated. We have the nitrogen protonated as well. So that's the first pKa. This is usually around neutral. The carboxylic acid is deprotonated. The N is still protonated. Because the pKa of a carboxylic acid is around 2, a pH of 7, it's going to be deprotonated. So only the amine here is protonated. At basic pH, everything is deprotonated. You don't need to memorize pKa values here of the amino acids and the side chains. The pKa of an amino acid carboxylate is around 2. So around 2, that's definitely going to be deprotonated. And all of the amines are around 9 to 10, so those will be protonated. And some of the side chains, if it has a different group on it, will also have associated pKa's. So again, you don't need to memorize side chain pKa's, but you should recognize from the functional group whether it's going to be protonated or deprotonated at certain pHs. And this may be something in biochemistry that you may have to memorize, but in this class you don't need to do that. We're just taking a plenary glance here. Amino acids usually appear as zwitterions, and a zwitterion is amino acid that has a positive and negative charge. So it's usually drawn as the CO minus and NH3 plus. This is the zwitterion. And amino acids are also amphoteric, which means they can act as acids and bases. So the OH minus here can grab a proton off of the amine group. And we're left with NH2 and the OH minus here plus water. Or the carboxylate oxygen can grab a hydrogen and become protonated. So in the negative form or this positive form, neither of these are now zwitterions because they don't contain both of the charges. So we're going to move into peptides. Glutathione is an example of a tripeptide. There's 3 amino acid residues that combine together with amide bonds to form glutathione, the tripeptide. So this is 3 amino acids. They are forming amide bonds. Which are also called peptide bonds. So glutathione is a free radical scavenger in your body. It can destroy free radicals that react with enzymes and other proteins. A peptide, the order is very specific here. In this particular example, we have glutamine, cysteine, glycine. If we switch that order around, that's going to change the structure and the identity of that tripeptide. So the way that amino acids are connected is important to the peptide structure. So if we have 2 amino acids, alanine and glycine, these are going to combine via loss of water. So it's a condensation reaction. This is like an amide coupling that we did with DCC. We form an amide. This is alanine glycine, where the amino end is here and the OH is on the end. So this is the N terminus. This is the C terminus. So this structure is different than glycine alanine. So the order matters. This is a peptide. The amino N is, again, the N terminus. This is the C terminus. And this would be the structure of it, kind of laid out flat here. Only the bonds that are in green can experience restricted rotation. And that is because the N and the double bond carbon are in resonance with each other. Okay, so in chapter 14, we learned that thiols can be oxidized to form disulfide linkages by the same process. Cysteine residues, which have the thiol group here. Cysteine residues in a protein can connect one peptide or protein to another through a disulfide bridge. So this is the disulfide bridge. Disulfide linkages can occur intramolecularly or intermolecularly. So if there's a disulfide bridge within the same chain, this is an intrastrand disulfide bridge. If you have a disulfide bridge between two different strands, this is an interstrand. Any of these disulfide linkages will affect the 3D structure and the function of many proteins, such as curly hair. Curly hair is made from disulfide bridges. Okay, so these are two sequences here of leucine enkephaline and methionine enkephaline. They differ by one amino acid. Both of these function in brain control of pain. And then we look at vasopressin and oxytocin, which only differ in a phenylalanine and an arginine versus isoleucine and leucine, and they have very different functions. Vasopressin causes reabsorption of water by kidneys, and it controls blood pressure, while oxytocin is what induces labor in pregnant women and stimulates milk production. So changing these amino acids changes their structure and their function. So in peptide synthesis, we couple a carboxylic acid with DCC, which we've seen previously. This is dicyclohexocarbodiimide, and that will give us a peptide bond. All right, and you can kind of see the mechanism there. It's cut off a little bit, but that's okay because you don't have to worry about knowing the mechanism. We've seen this reaction before, so this is a common reaction done to synthesize peptides in the lab. DCC is one of the coupling reagents that can be used, but there are others possible. So even though we can use the coupling, that DCC can't give us selectivity necessarily. The order of things can get tricky. So if you have R1, R2, we can add things in orders that we don't want it to add. So you can get R1 added to R2, R2 added to R1. We can get two R2s or two R1s. This is kind of like the mixed aldol scenario where you get a lot of different products. If we're just making one thing, that's fine. But for a peptide, that gets kind of challenging, especially with all the functional groups. So what we can do instead is use synthesis strategies we've used before, where we can protect the group. So we can put a protecting group on the amine and the protecting group on the carboxylic acid that we don't want reacting, and then do the coupling, and then we can remove the protecting groups. So there are 4 levels of protein structure. We have primary, secondary, tertiary, and quaternary. The primary structure is just the sequence. So that's just the sequence of amino acids. So you can see this is the primary sequence of human insulin. We can see that there's 3 different disulfide bridges. One of them is intrastrand, the other 2 are interstrand. The next level of protein structure is secondary. And you can have beta pleated sheets, and these are held together by hydrogen bonds. So this one is the beta pleated sheet. And that looks like if you made a paper fan where you fold it back and forth, and you can see that there. And there's hydrogen bonding occurring between these groups along there, those carbonyls. And then we have the alpha helix, also held together by hydrogen bonding. And those appear along there. To simplify structural representations of protein molecules, these are used in shorthand. So if there's an alpha helix, it uses this spiral. And for beta sheets, it's usually just a flat arrow. And then if there's coils in between that have no shape, then there's not enough associations or hydrogen bonds to form either an alpha helix or a beta sheet. So I forgot two important slides here, and this has to do with tertiary structure of protein. And the tertiary structure is the 3D folded protein. There's a couple of different types of interactions that are responsible. We've already seen the disulfide bridge. We can have ion bridges or salt bridges. And that is a positive-negative interaction between the side chains. So aspartic acid and lysine can have that interaction. We can have hydrogen bonding between side chains. And then this is a hydrophobic interaction. We have phenylalanine and leucine. These are our nonpolar amino acids. And then we can also have metal coordination. So two of these aspartic acids can be coordinated together via a magnesium or some other metal ion. So these forces that are involved here, if you have a whole bunch of these carbons around each other, this is the hydrophobic. Over here we have the disulfide bridge. This one here is a hydrogen bonding. And this is the salt bridge. Okay, so a quaternary structure results from multiple chains of peptides that are bound together. This one happens to be hemoglobin. So the four different colors, we have the green, blue, red, and yellow. Those are different tertiary structures that combine together to form this large quaternary structure. So enzymes catalyze almost all biological reactions, and a lot of these enzymes are made of proteins. And enzymes allow reactions to take place very quickly, which otherwise would take millions of years to complete. They also stabilize transition states. So a serine protease, which is shown here, is an enzyme that cleaves peptide bonds. So a serine protease is a class of enzymes. This one happens to be trypsin, which is a typical one. So this big giant thing is responsible for cutting up a whole bunch of peptides. This is a mechanism of a serine protease. This is referred to as the catalytic triad, where we involve serine, which is here. This is serine. This is histidine. And this is aspartic acid. So those are the three amino acids involved in the catalytic triad. The histidine and the serine are kind of acting as bases, and the aspartic acid is acting as the acid. And you can look through this mechanism. Again, this is not something that you need to memorize, but at least you have seen it when you go into biochemistry, and it maybe will look familiar then. In the first step here, we have a peptide that can enter into the serine protease. The nitrogen from the histidine can grab that proton from the alcohol in the serine, and this alcohol can then act as a nucleophile and attack the carbonyl of the amide. And now we form this tetrahedral intermediate here, and that double bond will reform, and then it will kick off the amine. The amine will grab the hydrogen and give it back to the histidine. And now we've lost the NH2 And then the histidine can grab a proton off of water now that was lost. And then that water can attack the oxygen, get another tetrahedral intermediate, and then we free up the carboxylic acid part of that molecule. So this is the catalytic triad, how it kind of works there. And again, this is a mechanism that you don't need to memorize. This chapter is kind of to give you a preview of biochemistry so that you've seen some things. And when you're taking biochemistry, you want to go back to your organic chemistry roots, which will help you to not have to memorize as much if you can understand the mechanisms within that. So that is all the content for the semester.